Dr. Robin Martin, welcome to the Coral Classroom. Come on in.
Hi, Greg. It's really great to be here.
This is Dr. Robin Martin. She's with the ASU Center for Global Discovery and Conservation Science. And if I get it right, you're also with the School for Geographical Sciences and Urban Planning.
That's correct.
What's a geographer and urban planner doing down here on the seafloor?
Well, geography has a lot to do with many things throughout the world, and there are corals everywhere. So.
Well, before we get into corals, we're here in Hawaii on the Kona coast, and I'm just curious, how long have you been working here in Hawaii?
I've been working in Hawaii for about 20 years now, both on land and in the ocean. In the oceans, I study coral color and health and how they grow.
That's pretty cool. Okay, let's talk about coral. So this is a pretty neat patch of coral. Looks like different colors. What gives these corals the brown color or this kind of pale green yellow color?
So the main coloring in a coral is from a single-celled algae called zooxanthellae, which means yellow-brown, that has chlorophyll in it, that captures light, that the algae turns into sugars that feed the animal that is actually the coral polyp. So inside here, there's an animal, and then it has the little algae all around the edges of it.
So our viewers will zoom into one of these polyps from another video clip, but I'm looking at it right here, and I see all these little reticular patterns. Is that the animal? What is that?
This is actually the skeleton makes the shape, and inside that skeleton there's what's called a polyp, which is the animal, and then on the outer edges of that polyp there's the algae, which is photosynthesizing, which taking light just like a plant does on land and turning it into sugar that it feeds to the animal. And in return, the animal gathers up other nutrients, and little animals that come by, it will gather them up and eat them and feed back to the algae the other nutrients it needs to make protein.
So let me get this right. So the algae is harvesting sunlight, just like any plant. It's doing photosynthesis, and from that light, it's converting literally sunlight into sugar.
Correct.
It's using that to feed the animal, is that right?
Correct.
And then the animal is doing other things, whether it's collecting other creatures that come by somehow or nutrients from the seawater and feeding that algae in return.
Correct. A process called symbiosis.
That's really cool.
Yeah.
So what is it that gives this one kind of a brown color and this one more of a yellow or yellow-green color?
The algae is the dominant pigment with the chlorophyll in it, is the dominant pigment that gives it its brown-yellow colors.
Okay.
But there are a number of other pigments that corals have. Just like in leaves, they have carotenoids, and there's also a brown pigment called phaophytin that they have. And then there's another set of pigments called reflective pigments, where those are the red and purple colors that you might see, and the blues, because those pigments are absorbing in one wavelength, and it creates a bluer color, like some down there are a little bluer, but sometimes you'll see a whole coral head that's blue.
That's really cool. So the colorfulness of a reef, at least the coral part of the reef, you're saying it's driven by both the coral animal itself and these proteins, but also by the algae that it has a symbiosis with. It seems like the algae kind of makes the coral more brown and yellow, whereas the animal makes it more blue and red and other colors. It's like a real kaleidoscope of colors coming together.
Exactly. And it's sort of the relevant It is the relative amount of those pigments that make the different shades.
So, Robin, your work in the ocean and on these reefs is really focused on the color of these corals. How do you do it? Is it work that you do in the water alone, or do you take some of this back to the laboratory to understand it?
Yes. So to understand the amount of pigments in the coral, I have to actually take little teeny samples and grind them up and then put them through what we call a spectrometer, which will measure the absorbance of the chlorophyll. Just like it's absorbing the light when it's in the spectrometer, I shoot it with light and you see how much it absorbs, and that tells you the concentration of the chlorophyll in the sample. And then I relate that back to the area of the sample that I took.
Well, Robin, I want to thank you for joining me here on this beautiful coral reef along the Kona coast of the Big Island in Hawaii. I really appreciate you joining the Coral Classroom.
Well, thank you so much for having me, Greg. It's beautiful down here.
All right, well, I'll see you around on the reef.
See you out there.